Good morning. This is episode 5 of the Antimicrobial Resistance Talks, or in short, the AMR Talks, where we're bridging gaps and sharing solutions. This podcast is an initiative of the AMR Research and Work on Innovation and Sustainable Solutions Research, also known as the AMR WISE Research Group from University Malaya. Our aim is to increase awareness about antimicrobial resistance. Now, you might be wondering, what is the big deal about antimicrobial resistance? Well, imagine a world where the antibiotic we depend on to treat infections suddenly become powerless. Picture a future where simple surgeries, once routine, become life-threatening due to the risk of untreatable infections. This isn't science fiction. It's a reality we're inching closer to every day. Antimicrobial resistance is a silent crisis that stretches across the globe, affecting hospitals, communities, and the very core of modern medicine. In this episode, we're pulling back the curtain on AMR and its journey through the microscopic battlegrounds where bacteria and antibiotics clash, and we'll meet the brilliant mind working tirelessly to find solutions through bacteriophages. In this episode today, we will be discussing bacteriophages, the challenges and the future it may bring us. We have today with us Dr. Tobi Nagel. She is the founder and president of Phages for Global Health, a non-profit organization that facilitates the application of phages in Africa and Asia, where approximately 90% of the deaths from antimicrobial resistance are expected to occur. Tobi previously spent 15 years in the pharmaceutical industry co-developing drugs that have been tested in over 80 clinical trials. Her highlighted achievements include being a Fulbright Global Scholar and serving as an ad hoc reviewer for the U.S. National Institute of Health Special Panel on Bacteriophage Therapy. She holds a PhD in medical engineering from the Harvard-MIT Division of Health Sciences and Technology. Good morning, Dr. Tobi. Thank you for joining us today.
I'm pleased to be here. Thank you for having me.
All right. So, phages, as I understand, are viruses.
Mm-hmm.
That sounds scary.
It does sound scary, doesn't it?
Yes.
But these are viruses that only kill bacteria. They don't do any harm to mammalian cells, human cells, animal cells. And they are actually already in your body and in my body. They are part of our natural microbiome.
How do they interact with bacteria?
So they've been evolving sort of against each other. This arms race, it's often referred to, where phages will attach to a given bacterial cell, inject their DNA, hijack the bacterial molecular machinery so that they're now making phage DNA instead of bacterial DNA. They make new phages within that bacterial cell, and then in the end, the phages use an enzyme to pop open the bacterial cell, thereby killing the bacteria, and in the process releasing a whole bunch more phages that can go on to kill nearby bacteria. And maybe for one phage that attaches to a bacteria, you might have 100 phages that pop out. 30 to 60 minutes later, it happens pretty quickly.
Wow. And all this happens without affecting us human cells.
Exactly. Because really they're only attaching to and killing and having this impact on the bacterial cells, not on human cells.
I see. Hence the name bacteriophage.
Which means bacteria eating, essentially.
Mm-hmm. So how do bacteriophages differ from traditional antibiotics when it comes to combating bacterial infections?
Yep. So traditional drugs are typically small molecules, or slightly newer ones are biologics like antibodies. And phages are a virus. And some argue, are phages truly alive or not? Because really all they are is DNA packaged in protein. And they can't really survive without hijacking a bacterial cell. But phages and bacteria are constantly evolving against each other. in this kind of evolutionary race. And so it may be that the bacteria evolves in a human, and we can actually use that for our benefit. I could talk about that a little bit. And the bacteria will also be evolving over time, so they have to sort of adjust to each other.
Oh, that's like genius packed into.
It is. And let me just give one of those examples that some researchers have found, especially at Yale University in the US, that you can, if you understand the molecular mechanisms of how phages and bacteria interact, in this case in terms of efflux pumps that bacteria use to spit out any antibiotics that might be used to try to kill the bacteria, you can use the phage to evolutionary push the bacteria so that it becomes sensitive to antibiotics again, which is a game changer in terms of our antibiotic resistance crisis.
That is very interesting.
Mm-hmm. So it's been done in vitro, but it's also been done in patients to use a single phage, and patients become sensitive to antibiotics again if you target that phage with an understanding of the molecular mechanisms.
I see. And how long would this process take?
I believe that's taken days or weeks in patients, so it can happen relatively quickly.
I see.
Mm-hmm.
Could you maybe share us any success stories or notable cases where phage therapy has particularly helped in treating resistant infections?
Yep, to my knowledge, there's at least 2,000 so-called compassionate use cases around the world, largely in the US, Europe, and Australia, where patients had no more options. Antibiotics were no longer working for them, and they were treated with phages. Many of those were quite successful in dramatic ways. So one of the more famous ones happened in San Diego in California, where a man had become infected and there was nothing else they could do. They were expecting that he was dying. And his wife, who happened to be a global health professor, set about saying, hey, as a last-ditch effort, can we try phages? She sent out a tweet, Dr. Stephanie Strathty, and my understanding is that within about 3 weeks, the global phage community of experts had found several phages actually, that worked against his particular bacteria. They had produced enough of those phages to treat him. They'd gotten emergency approval from the U.S. Food and Drug Administration to treat that patient, and he turned around, and he is alive today. And so they wrote a book called The Perfect Predator, the husband and wife team, as a way to start sharing this story of this is an important, potentially, and in his case, life-saving technology. They also have a new phage center at UCSD that grew out of that. Yale University is the one that's done a lot of the using phages to resensitize bacteria to antibiotics. They have their own ongoing clinical trial. Mayo Clinic has a number of these similar types of stories where perhaps someone was about to lose a leg to amputation due to a prosthetic joint infection, and they were able to turn that around and save the leg with phages. So Mayo now has a number of clinical trials either ongoing or about to begin.
I see. That's a whole new trajectory to combating bacterial infections. For all our listeners, we will attach the book.
Perfect Predator.
Yes, The Perfect Predator.
I might also add that there's a movie, another book called Salt in My Soul, a documentary was made out of that book, and it's about the story of a cystic fibrosis patient who sadly, unfortunately, did pass away. They started phage therapy, I believe, as a last-ditch effort about 48 hours before she passed, and later discovered that it was starting to work, but they had gotten the phages to her too late. So there are a number of really powerful books and also this movie out there.
There's one thing I was reading up, and I've seen that phages have actually existed before antibiotics.
And were used even before antibiotics. So In the U.S., Europe, and Asia, phages were starting to be used. I would say even in the 1917, I believe, is when that started, but especially in the 1920s and 30s. And then penicillin became available. And being broad spectrum, an antibiotic was easier to use. And so, in the 1920s and even 1930s, phages were being used in patients. I believe in India they were even used to sort of clean up well waters for cholera, that kind of thing.
Oh.
But when penicillin started being broadly used, more in the 1940s, I believe, without diagnostics back then, it was easier to use a broad-spectrum antibiotic, and it's worth mentioning here that phages are more, you can think of them as a more narrow-spectrum antibiotic, so they might only work against cholera or Shigella or E. coli, something like this, instead of an antibiotic that might kill several of those types.
So all this seems like a ray of hope in terms of antimicrobial resistant infections. But how about challenges? Are there any challenges or limitations that researchers face when we're working with phages?
Yes, a number. And some of those that are usually highlighted by phage experts are manufacturing and purifying phages at what's typically called good manufacturing production, GMP. Or regulatory issues, because as you can imagine, since we talked about phages not being your typical type of drug, it hasn't always been totally clear how phages fit into an existing drug regulatory system in each country. And a number of countries, especially in the US, Europe, and Australia, are starting to clarify how phages can fit in, and clinical trials are ongoing there, but that's been a process. And that process, I would say, has not really begun in most African or Asian countries. So that's actually what I'll be focusing on with the Fulbright Award you had mentioned. I'll be sharing information from the U.S., Europe, and Australia with specifically national leaders here in Malaysia and in Kenya and Uganda, so that we can help hopefully fast track how they choose to regulate phages in their countries.
I see. So, as you're mentioning Asian countries, are there any insights that you think would be particularly valuable for the people in Asian countries when introducing or implementing phage therapy?
One thing I will say is that public engagement, so communicating exactly how we're doing now, is really important for all of the stakeholders to understand about phages. So not only the scientists who might be developing or the clinicians who might be using them for treatment, but the patients, so that there's a general understanding. Because you're right, this idea of giving you a virus to treat your bacteria sounds scary on the surface until you start to understand how powerful and valuable it could be. I do wonder if the tendency towards, in many Asian countries, towards wanting to use natural products might favor people being more open to phages, perhaps, than other countries in the US or Europe. I also am sad to say that I think the desperation in terms of the number of people who we expect will die in Africa and Asia as compared to the rest of the world, I think that desperation will drive a need and a desire to adopt these new innovations like phages sooner than perhaps in other countries. I would love to see phage therapy take off faster in Africa and Asia than in, say, the US and Europe. I think that I often liken it to cell phones versus landlines, how many lower and middle income countries sort of just leapfrogged over landline phones and went straight to cell phones. I would love to see that happen with phages and phage therapy, that just leap forward and start using phages more broadly in Africa and Asia, because the need will be greater here.
Mm-hmm. In a previous conversation we had, you mentioned that in Africa, you sort of introduced phage to the general public. How is that like?
Yeah, and so to give a little more context for that, so we've been running, my nonprofit organization, Phages for Global Health, has been running laboratory training workshops in Africa since 2017, where we teach scientists, clinicians, students at all levels there how to isolate phages and begin to develop them as phage products. And then out of that has grown about 50 research projects that they have been working on, and they've gotten over $1.5 million in funding to take those projects forward. And as part of that funding, they have done more public engagement activities. So one of our projects that I was involved with is developing phages to decontaminate poultry meat in Kenya. And so as part of our funding for that project, the team in Kenya met with, I believe, over 350 poultry farmers to talk with them about phages and say, here is this new technology. What are your concerns? Would you use it? Would you be more likely to use it in the chicken's water or in the feed, so that we understand how we might need to deliver it? And so I think those types of activities, in whatever is appropriate, whatever context is appropriate for the intended application of the phage, whether it's poultry farmers or patients, it's going to be really important to continue to have those types of discussions.
Most definitely, yes. All right. And the reason we're having this conversation is because of antibiotic-resistant bacteria. So are there any concerns about the development of phage-resistant bacteria?
It's a really good question. And we do know that, as I said, there's this constant sort of evolution of phages against bacteria and vice versa. So we do know that bacteria can develop resistance to phages. But what's unique about phages as compared with traditional, say, small molecule antibiotics is there are a variety of tactics you can use to minimize or even reverse or almost get rid of that resistance developing. And so one of the things you can do is use a combination of phages. And we know that each phage typically binds, say, a receptor on the surface of a bacteria. And so the bacteria might mutate that receptor and therefore become resistant to that phage, being killed by that phage. But if you have many phages targeting that bacteria, and each of those phages bind to different receptors, then the probability goes way down that that bacteria could successfully evade the combination of phages. So that's often what's been done historically, is what's called phage cocktails, a collection of phages. Or what's being done more now that we understand more of the molecular biology underlying the phage bacteria interactions, maybe that receptor that the phage targets, you choose a phage that targets a receptor that's important for making that bacteria particularly dangerous. So I'm on the board of a company called Phage Pro that's developing cholera phages. And we have a 3-phage cocktail, and we know that each of those phages bind to different what's called virulence factors, or you can think of them as receptors on the bacteria that are important for making that bacteria very harmful to people. We know that if any of the bacteria mutate or evolve away from trying to avoid being killed by the phages, that's fine. It turns out those resulting evolved bacteria are no longer harmful to humans because we've targeted those so-called virulence factors on the bacteria. And so more and more of that type of knowledge is being built into phage drug development. So essentially it's directed evolution is what it's called.
Wow. That is really mind-blowing. That's the word I'd use.
Yeah, and the antibiotic resensitization that I talked about employs that same kind of idea, that same mechanism.
So getting all this done, I'm sure that you need particular infrastructures and very, very particular expertise to work behind all this. So from a practical standpoint, what are some of the things we need to implement phage therapy on a broader scale, maybe in healthcare systems?
Yeah, and let me start by saying that isolating a given phage against any bacteria that you want to focus on, that is typically quite easy. And if you can imagine, we mentioned that phage therapy was first done over 100 years ago. the labs were not that complex 100 years ago, and yet they were able to successfully isolate phages, purify them enough to give them to patients and see some results. So that those first steps of isolating phages can happen within days to weeks, and that's depending on the bacteria. Some bacteria are easier to work with than others. That's not that complicated, and that's what we teach through our laboratory training workshops. But then you do get into the drug development that requires more understanding of the dynamics between your particular type of bacteria and your particular phage. And I think it's critical to have a deep understanding of the infection that you want to treat. So maybe the body has ways of walling off. For example, in tuberculosis, there are granulomas that develop in the lungs. I wonder if phages can get across those granulomas, possibly not at that stage of the disease. But there are other stages of tuberculosis where phages might be able to access the bacteria. So you really need to understand how that bacteria is functioning or causing dysfunction in your body in order to do proper drug development, and make sure that you deliver the phage so that it actually can attach to that bacteria. So that might require different formulations, different microencapsulations, things like this that are a standard part of drug development. So it really takes bringing together phage experts with drug development experts and clinicians who understand the biology of the infection.
Mm-hmm. All right. And is there any ongoing research or innovations in phage therapy that you are particularly excited about?
So there, I think in general, working with natural phages and using these mechanisms we've talked about for directed evolution, or using phage cocktails, that in itself is a game changer. But then there are added technologies on top of that. So some groups are using CRISPR technology to enhance the ability of the phage, say, to deliver something else to the bacteria, a toxin or something that, let's say, that could kill the bacteria, in addition to the phage's own ability to kill the bacteria. So some of these double whammies, if you will, in terms of drugs, are pretty interesting. I had mentioned that at the end of the killing cycle, phages use a enzyme to break open the bacterial cell wall. Some of those enzymes are called lysins, endolysins, or lysins, L-Y-S-I-N. And more and more we're wondering if lysins themselves could be a unique drug, because they could be scaled up at a much cheaper rate even than phages. And phages are not that expensive to make, but lysins could be even cheaper. And lysins have even a lower resistance that typically develops against them. So that might be a new technology that we'll see coming forward as well. The first lysins first lysin is in clinical trials right now in the U.S.
I see. All right. You mentioned natural phages.
Mm-hmm.
Does that mean they exist around us? Is that what you meant?
Yes, phages are actually the most abundant organism on the planet. You've probably heard that there are many, many bacteria in the world, but there are even more phages than there are bacteria. And so that's why it's so easy to find phages that can kill a given bacteria. But then sometimes what happens is the phage might not quite have all the properties that you want it to have. So then people can genetically engineer them to have the properties that you want, either to make them safer or to make them more potent, that type of thing. So that's, and then there are different laws required, different regulatory systems required if you genetically engineer a phage. And that varies by country. So that's another factor to consider.
Mm-hmm. All right. So how do you think the scientific community and healthcare industry can collaborate to sort of further explore and develop phage-based solutions to treat antimicrobial resistance infections?
Yep, I think much of this work has already begun in academia, and it's quite easy actually to even have an undergraduate do their thesis on isolating a particular kind of phage and then characterizing it. then they do need to start partnering with some of those drug development experts who would typically be at a company. Or I don't know if in Malaysia there are government research institutes that are also doing some drug development. If so, those might be great partnerships. We have some support for a couple of our projects from GSK Global Health, which is the nonprofit arm of GSK, the pharmaceutical company. And GSK Global Health only supports projects that are intended for low and middle income countries. So it's exciting to see their enthusiasm for working on those. And in fact, we have 2 projects with them, one that's working on phages for a neglected tropical skin disease called Buruli ulcer that's mostly occurs in West Africa and a little bit in, somewhat in Australia. And then our other one is a licensed-based project that GSK Global Health is supporting. So organizations like GSK Global Health that are sort of a mix between a nonprofit and a pharmaceutical is often a nice way to help bring these products forward.
That sounds very commendable. Are there any specific bacterial strains or infections that are more amenable to phage therapy?
I would say that historically, we haven't mentioned that phage therapy has been used for over 100 years, largely in the former Soviet Union. That's when the rest of the world started using broad-spectrum antibiotics. The former Soviet Union continued to use phages. And so currently in the countries of Georgia, Poland, and Russia, they still use phages, and in some cases you can buy them over the counter.
Oh.
Yeah, in little vials. And many of those phages have focused on gastrointestinal infections, dysentery, Shigella, Salmonella, E. coli, those types. But they also have a number that could be used for skin infections, so you could put them topically on a wound, say a burn wound or something like that. They've even tried and have, I think at times, successfully used them for urinary tract infections. Anywhere you can deliver the phage in the body, theoretically, you can use phages. Mm-hmm. We're seeing there are about at least 30 clinical trials ongoing at the moment worldwide, and those tend to focus on, many of them are on cystic fibrosis patients who often get lung infections and have long-term exposure to antibiotics, so their bacteria become resistant. Just last week, Biomics, a company published successful clinical trial results in their cystic fibrosis patients, which is exciting. Several other companies are also working on cystic fibrosis. Some are working on gastrointestinal infections. Some clinical trials are for prosthetic joint infections or diabetic foot wounds that often don't heal very well. Those are some of the typical ones that are ongoing. Or ventilator-assisted infections that might occur. I believe there's at least one clinical trial ongoing for that. So it really comes down to if you can get the phage to it, either by injection, inhalation, IV injection, subcutaneous injection, or topical administration. Or swallowing. That's how most phages are taken.
All right, thank you for that. And in a broader spectrum, when we talk about EMR, will this fit into the comprehensive strategy to sort of combat AMR?
I think it's an important tool. I don't think phages will replace antibiotics. In fact, it's nice, often we're able to see some synergy, meaning that phages and antibiotics work better together than either works alone. So I think phages are going to be an important tool in the overall toolkit. And we really need a broad toolkit to overcome this huge problem. So antimicrobial resistant infections are expected to kill 10 million people by 2050. COVID killed 3.6 million people worldwide at its peak. And so this is a bigger problem that's coming. So we need a bigger arsenal of tools to overcome this. And that's really the purpose of Phages for Global Health. My organization is, many of those phage experts are not in Africa and Asia, but that's where the greatest antimicrobial resistance hit is going to be. So we're doing whatever it takes to facilitate building up that expertise in these countries so that hopefully phage products can be made in country rather than relying on having them delivered from overseas. And I will also mention that there's value in developing these phage products in country because, as we talked about, the local phages have evolved to kill the local bacterial strains. And so you're likely to have more success developing phages that you've isolated locally rather than phages that came from some other continent.
I see. That's very well put. Thank you so much. So for our listeners who would like to stay informed, are there any resources or publications or messages you would like to tell them?
So our website, Phages for Global Health, has a newsletter you can sign up for where we're regularly trying to send out more information. There's also an organization called Phage Directory that we work closely with. We've had some joint grants together, actually. And they have a newsletter called Capsid and Tail. So the capsid is the outside of the cell, and then the tail is the tail of the cell, if you will. So Capsid and Tail is a newsletter you can also sign up for, which has wonderful weekly updates on what's the latest in the phage therapy world, where are clinical trials doing well, where are positions open, where is funding possibly available. So I recommend signing up for either of those.
All right. Thank you so much, Dr. Tobi, for joining us today. I'm your host, Vedika Bhatt. This is the AMR Wise podcast series brought to you by University Malaya. This podcast series was created by Professor Sheila Srilasri Ponampalavanar, an ID physician, Associate Professor Dr. Cindy Teh Xuanju, a microbiologist in University Malaya, and myself, Vedika. To learn more about us, please visit our website mentioned below. This recording of the podcast was supported by the Department of Social and Preventative Medicine and the Visibility, Information and Communication Unit in University Malaya. Thank you for listening. Please join us and stay tuned for our next episode. This podcast is supported by a grant from the Foundation to Prevent Antibiotic Resistance. Till next time, bye-bye.
Bye.